Hi, welcome to my channel. Ever wondered how proteins finally fold into their native conformation or what role these secondary structures play? Do you know that these structures may serve as intermediate in the process of protein folding? We will discuss this issue in detail. So basically there are five secondary structures which are normally present in proteins, including alpha helix, beta sheet, beta turn, non-repetitive secondary structure, and the last one, number 5, is super secondary structure or motifs. The first one, alpha helix, has already been presented through a video, therefore this will not be discussed today. The remaining structure will be discussed and the emphasis will be on their physical features, stabilizing forces, and the significance. So regarding beta sheet, this is an extended sheet-like structure which is formed by pairing between two or more segments of a polypeptide or from multiple polypeptides. The structure appears to be pleated, therefore it is also called as beta pleated sheet, which simply means it is partially twisted. The hydrophobic amino acids are found in this structure more commonly, and they also play a role in making this structure rigid. The proline and glycine amino acid residues disrupt this structure because they are incompatible. So regarding the forces that stabilize this beta sheet, I will explain this with the help of this cartoon here. Here I have shown two segments which are arranged in an anti-parallel fashion. The size of these fragment or segments could be 3 to 10 amino acid residue long. Now, similar to any other secondary structure, this sheet is also stabilized by hydrogen bonding between carbonyl oxygen and amide hydrogen of the backbone or peptide bond, you can say. So peptide bond, the hydrogen bond is the main stabilizing force for the beta sheet. Hydrogen bond normally allow the binding of multiple segments, making this structure look like sheet with pleated surface and rigid structure. This rigidity is further complicated by the presence of hydrophobic amino acids. So under normal condition, this the multiple segments or clumps are okay, but when it happens excessively, it causes disease such as Alzheimer's, Parkinson's, and Huntington. In these conditions, these clumps are excessively produced and they accumulate in the cells, killing the cells, leading to disease. Therefore, clinically, these are very important in disease pathogenesis. Now, based on the orientation of hydrogen bonding as well as the orientation of the directionality of the participating segments, there are two types of beta sheet which I will discuss one by one in the next slides. So the first orientation is parallel beta sheet orientation. This is called parallel because the participating segments are arranged in a parallel manner or fashion. In this case, hydrogen bond is between carbonyl oxygen and amide hydrogen of the peptide bond. However, the hydrogen bond loses its planarity, and as a result of that, the bond is strained, it is weak, and this structure is rare under normal condition. However, in certain disease conditions such as Alzheimer, this orientation is more common and more stable. Now the second orientation, which is more common, is known as anti-parallel beta sheet. In this case, the participating segments are arranged in an anti-parallel fashion. This is more common, more rare, and the reason is that the hydrogen bond maintains its plane geometry. As a result of that, the bond is relatively stronger than you see in case of parallel. This structure is more common under normal condition. In this case also the surface in both cases will be pleated, and the rigid structure is the sheet like rigid structure. So coming to the second type of secondary structure, which is beta turn. This is called beta turn because historically this structure was identified as associated with beta sheet. This is also called as beta band or beta turn simply because it has capacity to change the direction of beta sheet in a protein molecule. Generally, they are present on the surface of a globular protein. Therefore, they are believed to compact the protein and help in protein folding. So what are the structure and the forces that stabilize beta turn? This structure is formed by 4 amino acids. Two of them are proline and glycine. They are most frequently present in this structure and they are believed to be responsible for the formation of this structure primarily. The remaining two could be any other type of amino acid. Similar to other secondary structure, this is also stabilized by hydrogen bonding between the first carbonyl oxygen and the third amide hydrogen of the peptide bond or the backbone. So this is all about the stability and the structure of beta turn. So now the third type of secondary structure, non-repetitive secondary structures. We have seen that the secondary structures such as alpha helix, beta sheet and beta turn, they are present in a polypeptide and their physical features is consistent or do not change in proteins. That is why they are called regular and repeating secondary structures. The remaining of the polypeptide is arranged in less regular structures. They are called as non-repetitive secondary structures. These structures are well defined, but they are not strictly regular or repetitive. It means their physical attributes vary in proteins. However, they are highly selective for a given protein. These secondary structures include loops and coils, but not the random coils. Like any other secondary structures, this type of secondary structure is also stabilized by hydrogen bonding between the backbone. So what is the significance of non-repetitive secondary structures? As I mentioned in the beginning that these structures may act as intermediate in protein folding pathway. Therefore, the first important function may be they play an important role in providing flexibility for protein folding process. And the second may be because of their role in protein folding, they are also important player in the biological function of protein as well. So coming to the last type of secondary structure, they are called motifs or super secondary structures. They are basically nothing but grouping of more than one type of secondary structure together. They are called as motifs. So this is specific recurring arrangement of secondary structure within a protein is called as motifs or super secondary structures. They are large, you can see, as compared to secondary structures, but they are smaller than protein domain. Protein domain we will discuss in later in a separate video. They often play an important role in protein structure and function. So this is all about the motifs or supersecondary structures. Biologically they are active. They may be more compact than the secondary structures. What is the significance of these? They play important role in various metabolic pathways. For example, in case of DNA metabolism, various DNA binding protein, they have these structures, motifs, such as DNA binding protein or transcription factors, or they also can play important role by forming channels in bacterial plasma membrane cell. They are called as beta barrels or porins, bacterial porins or channels. They make pore-like structures. This is mostly made up of the beta sheet. In humans, you will see the sodium channel, particularly voltage sensitive sodium channel, has 4 motifs, and in each motif there are 6 alpha helices which are present embedded in the plasma membrane. So this is also an example of the the motifs or supersecondary structures. So biologically they are very important as well. So finally, thank you. Please like it, subscribe it, and share with your friends, and let me know your comments. Take care and bye bye. One more thing, if you want full benefit, please watch it completely. Thank you very much. Bye.